Hello, my name is Sean Palecek. I'm a professor in the Department of Chemical and Biological Engineering. I'd like to congratulate you for being admitted to our PhD program, and I look forward to interacting with you as we go through this recruiting process. I'd like to take this opportunity to tell you a little bit about my research group. So first, a little bit about me. I actually grew up in Wisconsin, a small town in central Wisconsin. I did my education elsewhere before returning here as a faculty member. So undergraduate degree at University of Delaware, a master's at University of Illinois, a doctorate at MIT, and then a postdoc in genetics and cell biology at University of Chicago. As an undergraduate, I learned that I love to do research. I just like the challenge and the puzzle aspect of dealing with a really tough problem, and I love being on a university and really haven't left since. So I've been doing research for my career ever since an undergraduate. My lab works on the biotech side of chemical engineering. Specifically, we focus on stem cells and trying to take stem cells and make them into next generation therapies that clinicians can use to treat human disease. Our focus involves a lot of the biomanufacturing aspects of making cell-based therapies. My family still lives in northern Wisconsin. I spend a lot of time up there with my two boys here. This picture was not taken in Wisconsin, by the way. We go hiking, biking, fishing. Northern Wisconsin has a lot of lakes and woods, so we really like the outdoors up there. And during the pandemic, I've really upped my cooking game a bit, so I've been working on that quite a bit. I've been at UW-Madison for over 20 years, love the place. It's one of the few places I've visited that is really top-notch in engineering, biology, and medicine, which is important for the collaborative work we do. People around here really have a team-based spirit, and it'd be tough to find somewhere that is this collaborative in nature. This is a great thing for a student because you can be exposed to a lot of different perspectives, skills, and problems that you might not otherwise. But here's my research group. I currently have 10 PhD students working in 3 areas. 4 students work in the area of cardiac biomanufacturing. Here we try to turn stem cells into cardiac cells for use in vitro modeling, as well as for human cell-based therapies. So Jen, Martha, Aaron, and Fatima work in this area. We also have an effort in blood-brain barrier modeling using stem cells. So here we turn stem cells into cell types of the blood-brain barrier and use those in modeling development in disease as well as drug screening. So students working on this project are Ben, Koji, Feifan, Bill, and Sarah, and also have a project in antifungal therapies. In this project, we're trying to develop new peptide-based strategies to prevent and treat fungal infections in immune-compromised patients. So Doug works on this project. And our various research areas are also represented on this slide. A lot of our projects involve stem cell biomanufacturing. Here we try to take a stem cell and guide it through development to make cell types of interest. The stem cell we work on is the pluripotent stem cell. The cell type has 2 unique characteristics. First, it can divide an unlimited number of times. And second, it can turn into any cell type found in the body. So from an engineering perspective, we can make any cell type we want and as much of that as we want if we can figure out ways to do that. So we develop ways to turn developmental signaling pathway on and off. So the Wnt pathway here, we can use small molecule drugs to turn it on and turn it off at specific stages of the biomanufacturing process and make cell types of interest. This schematic shows our ability to guide cells through cardiac development to make endothelial cells, which make the blood vessel, the cardiomyocytes, which contract, fibroblasts, which I'll tell you about in a couple of slides, and smooth muscle cells along with the epicardium. We then take the cell types making these biomanufacturing strategies and apply them in cardiovascular tissue engineering. So we want to assemble these different cell types into a functional tissue. So here we've made cardiomyocytes from green fluorescent protein expressing stem cell lines so they glow green. And you can see that these contract as a tissue. I'll tell you a little bit about how we can incorporate fibroblasts into these aggregates to improve their structure. At the cell level, the cardiomyocytes look like this. They're block-shaped cells, and we've stained here the sarcomeres, which are the contractile elements. So you can see that these cells have the appropriate contractile elements in each cell, but that they can interact with each other in this functional tissue way. We also apply this stem cell biomanufacturing approaches to making models of the blood-brain barrier in collaboration with Professor Eric Schuster. So again, we guide stem cells through brain developmental pathways to make these endothelial cells. And then we can put these in a transwell model. So on the top, we can put a drug. On the bottom, we might put neurons, and we can study transport of the drug across this endothelial barrier and its effect on the neurons. And finally, we have this project with Professor Lin on synthesizing and delivering new antifungal compounds. So my lab works on making peptides that can specifically kill fungal cells without killing human cells. We use Professor Lin's layer-by-layer polymer film assembly to coat medical devices such as a catheter, load these peptides into the catheter, and deliver these in animal models to rats which are exposed to a fungal pathogen, and ask, can we prevent colonization of these catheters by fungi? So without the film and peptide, you can see this catheter has this biofilm of cells on it. When we have the layer-by-layer polymer film and the antifungal peptide, you have a clean surface. We can, at least in the animal models, prevent the biofilm formation. It's a very highly collaborative work. Here I'm going to show you collaborators outside of the department. I already mentioned we work with Professor Lin and Schuster in the department. We've had a long-standing collaboration with Tim Camp in the Madison School of Medicine. Tim's a cardiologist, studies electrophysiologies of the cells. So we collaborate to look at the electrical activity of the cardiac cells we make. Professor Inga is in cell and regenerative biology. She's a mass spec expert. We're working with her lab to investigate the metabolomic and proteomic profiling of our cardiac cells and comparing those to native cell types in the human heart. We will also work with Professor Akihiro Ikeda in UW-Madison Biochemistry to benchmark our cardiomyocytes with aging cardiomyocytes in animal models. Here we use NMR metabolomics to compare our stem cell-derived cells with cells in aging rat models. And we work with Professor David Andes, who's in infectious diseases in UW-Madison, to look at animal models of fungal disease using our peptide films. In addition to these UW-Madison collaborators, we have collaborators across the country and even around the world. Here I'm just highlighting three that we're currently working with. One is Professor Brenda Ogle at University of Minnesota. Brenda's lab performs extracellular matrix metabolomics. We look at the matrix or scaffold that the cells we make secrete, and I'll tell you a little bit about that with our fibroblasts. We collaborate with Professor Todd McDevitt, who's at the Gladstone Cardiovascular Institute and UC San Francisco. Todd's group has developed ways to take our 2D differentiation processes and perform them in a 3D bioreactor so we can make billions of cells that a human patient might need. We just started a new collaboration with Professor Ramani Ramchandran at the Medical College of Wisconsin in Milwaukee to study the role of cilia in vascular development in our blood-brain barrier model. And this is just a subset of some of the collaborators that we work with. Right, so now I'd like to give you just a little bit more depth into our recent unpublished efforts to make stem cell-derived cardiac fibroblasts. So this effort was led by Martha Floy, who's a fourth-year PhD student in my lab, and a collaboration with people I showed you in the last slide. So Tim and a scientist in his lab, Jin Hua, Todd McDevitt and a former graduate student in his lab, Orion Mathis, and then Brenda Ogle and Sophie Gibbons, a graduate student in Brenda's lab, all contributed to this work. So Martha developed a way to make fibroblasts, specifically cardiac fibroblasts from stem cells. A fibroblast, you can think of this as a support cell, so it makes the scaffold that comprises the heart in this case, and it also secretes factors that keep the muscle cells, or the myocytes, happy and healthy, and it allows them to respond to stress. There are actually more fibroblasts in your heart than there are muscle cells or any other cell type. In the developing heart, the fibroblasts come from two sources indicated by color here. So in your left ventricle and parts of your atria, they come from the primary heart field, and in the right ventricle and parts of the atria, your fibroblasts come from the secondary heart field. So Martha developed ways to guide our stem cells through primary heart field progenitors to epicardial cells to fibroblasts in a 3-week process by manipulating Wnt signaling and TGF-beta signaling, as well as fibroblast growth factor. Genois in Tim's lab determined a way to make the second heart field cardiac fibroblasts using a basic fibroblast growth factor signaling. So now we have these 2 cardiac fibroblast populations, and the paper we've been working on compares and contrasts those. So one thing we did in collaboration with Brenda Ogle was profile the matrix that was secreted by these, and I'm going to focus on Martha's cell types here. So we used matrix proteomics and found that our first heart field derived stem cell derived fibroblasts are very similar to fetal heart tissue, or the fibroblasts in fetal heart tissue, which is to be expected given the immature stem cell derived nature of these cells. We also found that Martha's cardiac fibroblasts will respond to stress. So if we apply serum to the cells, they'll upregulate this protein smooth muscle actin, which makes the cells more contractile. And in collaboration with Todd's group, we made these 3D aggregates in a fluorescent calcium reporter line. So with each flash, an aggregate is contracting, and we can monitor the rate of calcium release from the intracellular stores and uptake into those stores through image analysis. We can plot out various parameters here. One of the important parameters is the slope of this part of the curve, the Vmax up, or maximum upstroke velocity. What we found is if we have the stem cell derived fibroblasts, these aggregates handle calcium faster than without the fibroblasts. So that these fibroblasts are improving the function of these cardiac tissues. So our overall message of this story is that our advances in making fibroblasts from stem cells give us higher model fidelity of the cardiomyocytes, and we think that this will improve the ability to use these cardiac tissues for in vitro drug development and for in vivo cell-based therapies. To close, I'd like to also highlight one additional opportunity in working in my lab. So I'm the research director for a National Science Foundation Engineering Research Center on Cell Manufacturing Technologies. This is a multi-institution center. We have 4 major partners at UW-Madison, Georgia Tech, University of Georgia, and University of Federico Maguas, and then other domestic partners and international partners in Canada, Ireland, and Japan. CMAT has 50 graduate students and 80 faculty and an industry consortium of 20 industry partners. Our goals are to advance the cell therapy industry, make it easier for industry to develop curative therapies based on mammalian cells, including stem cells and T cells to fight cancer. Students who participate in this center can take classes with participants at other institutes. We have numerous collaborations across the center. So in addition to what I talked to you about, we have collaborations at Georgia Tech, and University of Puerto Rico. We have exchange programs where students can spend a few months to a year at these different sites, and we work closely with industry. So we have funding and collaboration with a number of industry partners here. We also have participation in a number of centers at UW-Madison, so departments in biomedical engineering, material science, the Cancer Center, Stem Cell Regenerative Medicine Center, collaboration with the Allen Institute of Cell Science in Seattle to advance the basic biology of cardiac cells from pluripotent stem cells. We work with Weissman Biomanufacturing to scale up some of our technologies. Thank you for taking the time to listen to this presentation about my research group. I look forward to interacting with you one-on-one throughout this recruiting process.